In today's video, we're going to be taking a look at meiosis for A-level biology. Now, before we jump in, let's quickly take a look at what diploid and what haploid means. So firstly, ploidy refers to the number of sets of chromosomes that an organism has. So in diploid, we have two chromosomes, two sets of chromosomes. Humans are diploid organisms. We have two sets of chromosomes. One set comes from our mother and one set comes from our father, and that results in us having in total 46 chromosomes. Haploid refers to just one single set of chromosomes of an organism. So in humans, the haploid number is N. Well, it will always be N, but the haploid number of chromosomes is 23. If 2N makes 46, N will be 23. If, for example, we have a tetraploid, which means that the ploidy number is 4, so it's a 4N, it's a tetraploid, and let's say that that results in 40 chromosomes in total. The haploid number, so just one set of chromosomes, one full set of chromosomes is N, all the time it's N, and in this case, if 4N is 40, N is going to be equal to 10 chromosomes. So that's diploid and haploid covered. Now let's take a look at meiosis. So in meiosis, we have two phases. We have meiosis 1 and we have meiosis 2. Now in interphase, what happens is we have our growth phase 1, synthesis phase, and then our growth phase 2. In synthesis phase, so in late interphase, the centrioles in animal cells are replicated so that we have 2. In plant cells, they don't actually have centrioles. So what they have instead is an mTOC, a microtubule organizing center. Now, before we continue, let's quickly cover what does microtubule mean, centriole mean, spindle and spindle fiber mean. What is the relationship between all of these terms? Well, centrioles organize microtubules. Microtubules form spindle fibers, which in turn make this 3D shape, which is called the spindle. Now, before going on to prophase 1 of meiosis 1, we need to cover what are homologous chromosomes. So homologous chromosomes are simply chromosomes that are the same shape, size, they have the same banding patterns, and they have the same genes at the same loci. Now, as we talked about before, humans have, for each chromosome pair, we have one chromosome from the mother and one from the father. And these chromosomes will be homologous. Okay, so for chromosome pair number 1, one will be from the mother, one from the father. Those will be homologous chromosomes. For chromosome pair number 2, one will be from the mother, one will be from the father. Those two chromosomes will be homologous. Okay, so homologous chromosomes are chromosomes that are the similar shape and size. They have the same banding patterns and they have the same genes at the same loci. Note, the fact that they have the same genes at the same loci doesn't mean that they have the same alleles, right? Alleles are variations of genes. So they will have the same genes at the same loci, but that doesn't mean that they will have the same alleles. So now let's take a look at prophase 1. We have those homologous chromosomes coming together and they will form bivalents in a process called synapsis. Those bivalents will then wrap around each other. So if we take a look here, we can see this shaded in chromosome will be our paternal chromosome and the non-shaded chromosome will be our maternal chromosome. So the non-sister chromatids, so this chromatid with that one or that one, and this one with that one or that one, will wrap around one another. And in doing so, they will produce a tension. Now, that tension can cause breakages to form, and those are called chiasmata. Now, where those chiasmata occur, there are exchanges of genetic information. And what we can see here is we have this initial paternal chromosome, but now it's got regions of maternal chromosome within it. Likewise, the maternal chromosome now has some paternal pieces of genetic information within it, right? That occurs as a result of prophase 1. And this process is called crossing over because genetic information is being crossed over. Now, before we continue to metaphase 1, let's quickly take a look at what does maternal and what does paternal mean and how we can remember it. So, maternal means that the chromosome is from the mother. Maternal just means it's from the mother. And the way that we can remember this is M for mother. Paternal refers to the father. P for papa or dad, okay? So paternal from the father, maternal from the mother. Now, let's look at metaphase 1. So the spindle fibers attach to the centromeres of the chromosomes, and they align them, well, the homologous chromosomes randomly orientate themselves at the cell equator. Now, this is another source of genetic variation, the random orientation of the homologous chromosomes. The chromosome can either, let's say this is the paternal chromosome, it could arrange itself on either the left side or on the right side, likewise with the maternal chromosome. Then at anaphase 1, the spindle fibers retract to their opposite poles, pulling away the homologous chromosomes away from each other to the opposite poles of the cell. Then we have telophase 1 and cytokinesis. Now, in telophase 1, in most animal cells, a nuclear envelope will reform, and cytokinesis will then occur, right? The microtubules will pinch together the cell surface membrane, and then a cleavage furrow will occur. In most plant cells, on the other hand, a nuclear envelope doesn't actually occur, and a cell plate, so in plant cells, a cell plate will occur. There won't be that cleavage furrow, well, there won't be the microtubules pinching together the cell surface membrane. So that's meiosis 1. In meiosis 2, the centrioles will replicate. So now we will have 4 centrioles. Before we just had the 2. Now we'll have 4. Note, genetic semi-conservative DNA replication doesn't take place. So just the centrioles are replicated, and now we are in prophase 2. The DNA condenses again back into its chromosomes. We then have the chromosomes aligning at their at the cell equator, and then the spindle fibers attach the centromeres. The centromeres then in anaphase 2 are divided in 2 as the spindle fibers retract and pull the sister chromatids to opposite poles. Okay, so the sister chromatids are now being separated to opposite poles. Cleavage furrows then occur. And then what we have is we have 4 genetically unique daughter cells from the initial parent cell. Now, what's very important to understand is that in anaphase 1, chromosomes, so full chromosomes, are being separated. The homologous chromosomes are being separated apart. In anaphase 2, the chromosomes themselves are being separated. So the sister chromatids are being pulled to opposite sides. So make sure you remember that difference, that in anaphase 1, the chromosomes are being separated. In anaphase 2, the chromosome, like the sister chromatids, are being separated. Now, it's actually very important to understand this little acronym, okay? PMAT. So it helps us remember meiosis, the stages of meiosis and mitosis. So we have P for prophase, we have M for metaphase, we have A for anaphase, and then we have T for telophase. So if you can remember PMAT, then you'll be able to remember all of the stages of mitosis and meiosis. Now, let's actually take a look at how meiosis results in genetic variation. So, number 1, it's that crossing over that occurs between bivalents. So homologous chromosomes, they form bivalents, they wrap around each other, that tension causes breakages at points called chiasmata, and then there are exchanges of genetic information between the homologous chromosomes. So that's crossing over. That's the first source of genetic variation by meiosis. We then have the random orientation of the homologous chromosomes in metaphase number 1. Okay, so the homologous chromosomes can arrange on either side. It's completely random. There is nothing. They're independent of one another. So that's the second way that meiosis achieves genetic variation. And then finally is that these genetically unique daughter cells from, let's say this is the father, will then go and randomly fuse with the genetically unique daughter cells of the mother. So that random fusion is the third and final way that meiosis provides genetic variation. So that's it for this video. If you found it helpful, take a look at some of the other content on my YouTube channel that might interest you. Otherwise, thank you very much for watching, and I hope you have a great day. Bye-bye.